So, yeah, all right. Normally, I get stuck rambling on about all of the background for far too long, but fortunately today, thanks to the previous talks, I can now ramble on about the experiment that we did, since you are now all expert in the laminar background and the anatomy and all the considerations that go with that when you're trying to think about how to put together a task to test with these functional measures that we were discussing. So, we heard a lot also about what the use of this would be for, say, discovery science, or what the interest would be for people who are interested in more cognitive science. So that is what we do. And I think that this is specifically the potential to assess connectivity between different regions right across the brain using the cortical layers is one of the main motivators for cognitive science in general to try to apply this technique. So I'm going to be talking about our work with depth-dependent BOLD to assess directed connectivity, not in visual cortex exactly, but we move a few centimeters up and we deal with the occipital temporal sulcus. So we use a reading task, but can talk about that in a little bit. So yeah, we talked previously about a number of these studies already, but in recent years there's been growing evidence that yes, indeed, there is some there is a strong spatial coupling between the hemodynamic response and the different cortical laminae that we can actually use to measure task effects in the deep layers or the superficial layers or the middle layers, depending on whether you're looking at feed forward or feedback. Specifically, that feed forward tends to focus on the middle layers or targets the middle layers, whereas feedback will target outside of the middle layers, so non-granularly. And we have mostly seen work in primary sensory cortices with this, so visual cortex, auditory cortex, motor cortex, and some work linking specific oscillatory frequencies to the different cortical laminae as well. But we go into a slightly more complex task where we look at language. So we do a single word reading task, right, presented in Dutch. We collected data on 22 subjects, and you can see a sample of the stimuli here. Presented. So the first two are real words. These would be salmon and battery in Dutch. The second two are pseudo words, so in red, right? And in the kind of fuchsia color, you have false font tokens. And the idea behind this is that the bottom-up information, the bottom upstream, the sensory information associated with all of these tokens will be roughly the same, right? And there isn't really anything different in the visual stream comparing, say, the superficial or the real words and the pseudo words, right? Because these are all legal orthographic strings that Dutch readers will be able to understand. The difference is that the real words will have meaning associated with them, right? The real words are going to have experience-based information, knowledge on the participant's part on this, and they'll be able to draw from all sorts of different information related to the lexical, say, word frequency, even emotional experiences with the words. So we're not specific about what the top-down information necessarily is so much, but we do make assumptions that it has to do with the semantic content of the words, the phonological content of the words, these types of things. So the idea is that by comparing that's too fast. Yeah, so the idea is that by comparing the difference between the real words and the pseudo words, we can actually have a strong effect where we only manipulate the top-down signal, where the bottom-up signal stays constant, right? And in doing so, we can then try to see what the effect of the top-down signal on the bottom-up signal actually would be. And then if we were successful in this, we were planning on going forward to assess the directed connectivity and seeing where this was actually coming from using GPPI, so the psychophysiological interaction analysis that many of you may already be familiar with. So, okay. The predictions that we had, and excuse this schematic, but I think it serves the purpose. So if you imagine you have two areas here, right, represented by these stained sections, and we would be presenting a real word or a pseudo word to a Dutch participant whose eye is here on this slide, right? The real word and the pseudo word would both hit the eye the same, right? No differences there. They would both travel back to these early visual areas, right? And then they would be taken into some unnamed lower sensory area, right, into the middle layers here, into the granular layers. Right, now from here, there's a whole mess of intrinsic connectivity that will be going on within the first region here, which would then project forward into a higher order region, and then ideally project back right into the original region. And so the idea is that the feedback from the higher order region to the lower order region would be stronger for the real word, which would be tafel in this case, which is a Dutch word. And that this would result in a higher amplitude, suspect or no, higher amplitude in the deep bin, right, which we could then use to test to see if we see a difference between these two words, right? And all this would be happening in the presence of a constant bottom upstream. So the middle layer, we didn't really expect to be manipulating, right? So what we did was we identified a region in the OTS, right, using a contrast of the real, the pseudo words, and the false fonts to pick out the region that responded well to the, say, orthographically legal characters, but then showed preference to the real and pseudo words over the false fonts, because this is a functionally defined region which sometimes goes by the name of the VWFA, the visual word form area. But we're not being specific about what we actually call it, right? So we identified this region, right? We would see how well you can see that. Yeah, so we identify this region, and then we generate our surfaces, right? And we perform an equivolume parcellation. I used Tim's implementation of it, but following the Weinert method, right? And we then demixed the signal from the different layers, right, using the spatial GLM, which was also implemented by Tim. Thank you very much. And registration tools implemented by Tim. Tim did very much work. But you can see here on the left column of the longer figure, right, if you look in green, in the green box, you can see magnified what the area we're looking at is. Right, and you can see in the middle window there, the middle cell, you can see what the mean functional image looks like. Right, and then in the bottom, you can see the ROI zoomed into that. Right? So you can see that the surfaces follow along very nicely. And the anatomy image there is an inversion recovery EPI that we took for the sake of registration. Right, we can talk about that later if someone's interested. And you can see on the right-hand side Yeah, and you can see on the right-hand side, going through from white matter to the peel surface, you can see how we did the voluming, right, with each voxel there that is yellow. More yellow has more of its voxel volume in a specific bin, and we go through the entire gray matter volume, and we assign each voxel to a particular depth bin, and this is what we use to actually look at the depth-dependent signal. So what we found was interesting. So if you, and I mean that in a good way, right? This that's a good interesting. So we had three gray matter bins that we were looking at, right? We had a deep, a middle, and a superficial. And as we talked about, we know that that's not the true anatomical picture, right? But we also know that bold, even sub-millimeter bold like we have, or sub-millimeter fMRI, is not going to be sufficient to really be able to resolve this finer anatomical structure. So we stick with three gray matter bins, and we know that, yeah, this is going to be integrating activity over, say, deeper layers, middle layers, and superficial layers, but roughly that the distinction should be preserved enough that we can tell differences in functionality based on differences in the input and output layers, right? So what we see here after testing for main effects of condition being the real word versus the pseudo word, right, for example, and depth, and then the interaction between the two, we see that the deep layer prefers very much the real word condition compared to the pseudo word condition, right? So you see this kind of, you see this effect where the deep bin will show stronger for real words, but higher for pseudo words in the middle and superficial bin. So it kind of reverses, right? And this is interesting because the overall signal for the pseudo word condition is actually lower, right? So there seems to be a specific deep bin. It's higher. Oh, sorry. The overall signal for the pseudo word that we observe in the non-laminar context is always higher, right? The real word is lower. So there is there's the assumption that there is a kind of inhibition that goes on, which is related to the top-down information in the say the semantic knowledge that you have of the real words, right? And so we can observe this here in the deep bin, which we predict will be the recipient of the feedback termination, the feedback signal, right? And seeing that the middle bin is actually higher for the pseudo words, the overall signal is higher for the pseudo words, we began to wonder if there was perhaps some kind of suppression effect going on that we could measure, right? With the to see if there was an effect on the middle bin from the deep bin, which varied with task, right? So we get to the interesting stuff, so I'll speed along. But we tested this in terms of a PPI, right, to see the effect of deep on middle. And we saw that deep and middle interact significantly during the pseudo word condition, but not during the real word condition, which is interesting considering that the overall signal is higher for pseudo words, but the deep bin signal is higher for real words. So we took this as some evidence of a suppression effect, perhaps, whereby the top-down signal acts to modulate the bottom-up signal and suppress the middle bin signal for the pseudo words compared to the real words, right? But I'll go through this. Yeah, I'll skip this one. This is the very interesting slide that I wanted to share with you guys today. So if we take the deep bin signal and we take the middle bin signal, the time courses that we extract from this slide here, right? We take time courses from the deep bin and the middle bin, right? And we create interaction terms with the tasks to see how they interact with the rest of the brain. We find that the deep bin almost surgically targets areas related to lexical retrieval, areas in the core language network, right? So you have posterior MTG and MTG here. And this is the only two clusters that survive correction. It's only on the left. The middle bin does not show any clusters really surviving correction, and it does seem to show preference for pseudo words, but we could talk about that later if you're interested. But the I think fascinating thing about this is that it seems that with gradient equibold you can actually disentangle very spatially localizable signals, right? And the GPPI appears sufficient to regress out task effects enough to actually let you observe the interaction between the middle bin and the deep bin in different areas of the brain, even though those regions are so close together. Right? So this is a very positive outcome from our work that we look forward to exploring in the future. So I guess we're done. And I thank you for your attention. Yes. Tom.
Yeah, it's more of a clarification question. Maybe it's different meaning. So, as I understood it, one of the things that spatial GLM does is kind of costs you space, right? You go from a signal that's like voxel by voxel, and then you sort of turn it into a GLM, which is giving you each layer as a separate time, and then am I talking?
Costs you space.
Can you comment on the spatial specificity of the findings? As I understood it, one of the things that happened when you put everything into this big spatial GLM is that you lost the ability to make spatially specific claims. Your last slide.
Well, you gain the ability to make claims going through the cortical volume, right? You lose the ability to make claims over, say, the surface, right? Yeah.
Okay, so then how can you elaborate a little bit on how you've ended up with this very fine, detailed spatial map that you showed at the end?
Fine detailed spatial map at the end. This one. Okay. Oh, so this is a the GPPI at the group level. So this is the group level map. This was done in normalized space. So I took so for every subject, the different bins always stayed in native space, right? And when I conducted the group level analysis, it was always to compare the bins and the native brains. So we predicted activity in the normalized brains, right, using the native space data, right? So that's why it looks. So this is a template map, right, that I had normalized and aligned every subject's brain to. right? But the actual measure that we used for the GPPI, right, the seed regions themselves, those came from native space, right? So that's why it looks like this. Yes.
That's actually
Oh.
the interest of time.
Sorry.